All right, just say welcome everybody back to our monthly Brainstorm Live session. I see some more college football fans, college sports fans, just cheering their teams on in the chat. So if you want, share your favorite university that you cheer for on sports days. Maybe we'll learn something about one another and find some points of connection. As a reminder for our format, we normally do our formal time for the first hour. And then after that, some of us might stick around for more informal conversation about, what was brought up for us, how we might apply some of the things that we learned about today from Dr. Bedrosian and Dr. Fonken. So without further ado, yes, and you do get CEs. You have to be here at the end. Obviously, to get those, you'll see a survey go out in the chat box, and we'll post that a few times at the end of the presentation. So without further ado, the floor is yours.
All right, hi everybody. Thanks for the invitation to speak today. It's great to be here with you virtually. My name is Tracy Bedrosdian. I'm an assistant professor of pediatrics at The Ohio State University, and I have a basic research lab at Nationwide Children's Hospital. And I'm here today with my collaborator. I'll let her introduce herself.
Hi, everyone. I'm Laura Fonken, and I'm an assistant professor at the University of Texas at Austin in the College of Pharmacy. And my lab studies neuroimmune activation and circadian disruption.
Great. So Laura and I have actually collaborated together for years and years, beginning when we were both graduate students together at Ohio State. So today we're going to talk a little bit about both some of our older research as well as some newer activity that we've been working on the topic of circadian rhythms and mood. So next slide, please. We have 4 learning objectives for you today. We hope by the end of the session, you'll be able to describe the circadian system, better understand how light affects the brain, identify sources of circadian disruption in the environment, and define some of the negative health repercussions of disruption to the circadian system. So the structure of today's talk is going to be divided up into 4 different parts. So I'm going to begin by providing a brief introduction to the circadian system, and then I'll talk about some of my past work investigating how exposure to light at night affects circadian rhythms, mood, and brain plasticity. And then in the second part of the talk, Laura will discuss how neuroinflammation serves as a potential link between the circadian system and mood, as well as some strategies for mitigating circadian disruption in our own daily lives. So I'll start with the introduction. So the word circadian, you might have heard it before, it comes from a Latin phrase, circa diem, which means about a day. And we use this phrase to describe biological variations that occur with a cycle of about 24 hours. So if you think about it, nearly all life on Earth is subject to this predictable daily cycle of brightly illuminated days and dark nights. And coinciding with this, nearly all organisms possess an endogenous 24-hour biological timekeeping system, which is called the circadian system. And that really just parallels this external light-dark rhythm. And, it really is virtually all organisms, ranging all the way from unicellular cyanobacteria up through humans that exhibit this type of daily regulation. So why is it important to have circadian timekeeping? Well, in practice, the circadian system coordinates a number of homeostatic functions, particularly in mammals, to ensure that biological processes are optimized for what's happening in the outside world. It's basically beneficial to be able to anticipate recurring environmental changes. And, therefore, having this circadian system can help an organism to maintain its optimal physiological function. When most of us think about the circadian system, the most prominent rhythm that comes to mind is, of course, the sleep-wake cycle. However, the circadian system controls many more aspects of physiology and behavior than just that. It's really exquisitely timed to be able to do things like anticipate food availability, for example, or the risk of encountering a predator, or availability of mates. Really, the list goes on and on. As humans, we can readily recognize many effects of the circadian system on ourselves. For example, of course, during the day, we have the highest alertness and the best reaction time. Whereas at night, our body temperature decreases, our digestion is suppressed. These are all things we can kind of observe about ourselves, but there's even much more than that going on without our awareness. So let me describe a little bit about the biological underpinnings of circadian rhythms. So in mammals, circadian rhythms are initiated in the brain within the suprachiasmatic nucleus of the hypothalamus, which is also called the SCN. And the SCN functions like a molecular pacemaker, sending out signals to help keep time within the body. And light is really the dominant entraining factor that synchronizes this internal clock to the external environment. And the way that happens is that light is detected in the eye by these specially intrinsically photosensitive retinal ganglion cells that are sensitive to light in the blue range of the spectrum. And that light information gets transduced through a pathway called the retinal hypothalamic tract directly to the SCN, which basically just tells the brain whether it's day or night. And I should mention, though, that although light is the most potent entraining factor, for timekeeping, the system is really very complex, and other recurring cues can contribute in other ways, such as daily temperature fluctuations, or even things like the timing of meals or activity. And so, although these circadian rhythms are initiated in the SCN, most tissues and cells throughout the body actually have the machinery necessary for self-sustaining oscillations. And these are so-called peripheral clocks, or extra SCN oscillators. And they're important for maintaining homeostasis within the local biological environment. So local rhythms can be influenced both by the central clock in the brain, as well as by other factors that are kind of specific to the local environment. Importantly for this talk, since we're talking about mood today, the SCN projects directly to mood-related structures in the brain, and it also regulates hormonal responses, including things like melatonin production via the pineal gland, which just helps to regulate peripheral clocks in the body, and also cortisol production via the hypothalamic pituitary adrenal axis, which you may know is a critical pathway that regulates stress responses, autonomic function, and has been really extensively researched in relationship to mood disorders. So I'm going to dive a little bit deeper into the kind of the biology, and this will be as deep as I'll go, so bear with me. But the molecular machinery that keeps time within the SCN is really based on what we call a transcription translation feedback loop. And there's a little refresher here on the left if anybody needs it. But essentially our DNA provides a blueprint of all the components that are needed to build the circadian clock. And from this blueprint, the relevant genes get copied on demand into RNA, which is the intermediate material that's needed to assemble proteins, which are the ultimate functional units of the clock. So like I said, the circadian clock is based on a transcription translation feedback loop. And so there's two key proteins known as clock and BMAL, which essentially bind to sequences in the promoters of other genes known as PER and CRY that activate their transcription at the beginning of the circadian day. And those gene products of PER and CRY accumulate, they form a complex together that translocates into the nucleus of the cell, and it interacts with clock and BMAL and represses their transcription. So the most important thing that you really just need to take away from this is that this molecular feedback loop takes about 24 hours to complete. So the daily timekeeping machinery is really in place all the way down at the molecular level. One more important thing to note about this is that this feedback loop not only regulates itself, but it also regulates other clock-controlled genes. And recent research suggests that up to 55% of protein coding genes in the entire genome are regulated by the clock. So really most aspects of physiology are sensitive to this timing. So what happens when the circadian system is disrupted, perhaps by aberrant light exposure, since we're talking about that today? Well, some examples will be very familiar to you. For one, seasonal changes in day length are known to alter mood. At these kind of temperate latitudes, like those of us who are currently in the Midwest, for example, about 10% of the population experiences winter depression, characteristic of seasonal affective disorder. And with that, symptoms tend to remit when the day lengths become longer during the summer months. And these symptoms are really thought to be related to a desynchronization between the internal circadian rhythm and then the external environment. In particular, the daily melatonin rhythm becomes shifted out of sync because of the lower levels of light during the daytime. And we know that things like bright light therapy in the morning can help to suppress that melatonin secretion at the right time and re-entrain the rhythm, which does improve symptoms in some patients. Jet lag is another example we're all familiar with, where rapid phase shifting causes light exposure to be out of sync with the biological rhythm. And experimental models of chronic jet lag have revealed really profound effects on brain function and behavior. For example, hamsters in a lab that are subjected to 6-hour phase advances every 3 days over a month develop really strong learning and memory deficits and reduced brain functioning. In human subjects, we know that flight attendants who are working transmeridian flights with really short recovery time between their shifts develop some level of temporal lobe atrophy, about 3% decrease in volume in one study, and cognitive deficits, so things like decreased reaction time and decreased correct response rate to different tasks. And then finally, an example that we're really going to focus on in this talk is exposure to light at night. And this one is really a pervasive source of circadian disruption in probably most of our daily lives, and it has the potential to affect multiple body systems. All right, so what are we talking about when we say light at night? Well, a little background. So for most of human history, if you kind of think of it in a grand scheme, before the advent of electric lights, humans were really exposed to minimal nighttime light. Full moon on a clear night illuminates the environment up to about 0.3 lux. And for comparison, basically a candle that's placed a meter away from you would only cast about 1 lux of light. At the end of the 19th century, with the invention of electric light bulbs, exposure to artificial light grew very rapidly, of course. For the first time, we could effectively artificially extend our days. And so night shift work was introduced soon after that. And, as technology boomed, humans have been exposed to even more sources of light at night, including things like television and computer screens, smartphones, tablet computers, etc. We know that over 80% of the human population and 99% of those living in the United States or Europe experience light pollution. So about 2/3 of Europeans and about 80% of North Americans cannot see the Milky Way due to artificial night sky glow. And about 15 to 20% of the population, by some estimates, is engaged in shift work. And of course, all of us are exposed to nightly light pollution from other sources. Just to give a few more examples for you, kind of the light intensity on an average urban street is estimated at 5 to 15 lux, and a typical living room could be 100 to 300 lux. If you have an electronic tablet computer in front of you, it emits about 40 lux, depending on the size of the screen. According to the National Sleep Foundation, about a third of adults and children actually sleep with an electronic device like a television or a computer screen on in their room. And about 87% of women report watching television in the hour before bedtime. I'm definitely guilty of that one. But just to say that this amount of nighttime light exposure is really unprecedented in human history. So how much does this light exposure really affect our biology? Well, we know from empirical studies that as little as 6 lux of light can suppress melatonin production in humans, which is far lower than, the kind of lighting you'd encounter on the street or, in your home often at night. And even lower levels of monochromatic light in the blue spectrum can suppress melatonin, which is really relevant because of this pervasive use of blue light emitting technologies at nighttime, like cell phones and things like that. So given the sensitivity of the biological clock, artificial light at night can really cause profound circadian and physiological disruption. One vulnerable group that I want to highlight is shift workers. So as I mentioned, 15 to 20% of individuals are engaged in shift work that exposes them to light at night on either a chronic or a rotating basis. And epidemiological studies have repeatedly linked shift work to symptoms of depression. Workers that are exposed to shift work are more likely to suffer depressive episodes, and prolonged shift work lasting more than 20 years has been associated with an increased lifetime risk of major depression. In one study that exposed student nurses to a single night of shift work, they were able to even detect a change in mood from that short exposure. And the graph here I'm showing is from a more recent study of non-shift workers, where they simulated a shift work environment by flipping them onto a 12-hour shifted work-sleep schedule. And this resulted in really rapid changes in the self-reported mood of these individuals. So reduced reports of mood and well-being. So this just demonstrates how powerful this circadian misalignment can be for mood. Slide. So another vulnerable group to highlight is elderly individuals in hospital environments or nursing facilities. So these individuals are particularly vulnerable to exposure to artificial light at night because 24-hour nursing activities and also safety concerns simply require a high level of nighttime lighting. Circadian disruption is already a factor in the natural aging process. We know things like the melatonin rhythm will decrease in amplitude, which is what you see on the leftmost graph here. And we know that sleep becomes more fragmented with age. And Laura's data on the right here showed that light levels in a hospital inpatient room fluctuate dramatically during the night, up to, almost daytime levels. So some large cohort studies have told us that exposure to light at night in these populations is associated with things like insomnia and poor sleep quality, as well as depressive symptoms. So light is a very powerful disruptor across all ages. All right, so that's kind of very high level overview, just kind of on the circadian system and how it's disrupted by light. I'm going to move into summarizing some of my own research on the topic now. All right, so I gave you some examples of associations between light at night and mood, but I'm going to talk about now what empirical studies tell us in the lab. So as part of my graduate work, we had set out to directly test the consequences of light at night exposure on mood and on the brain using hamsters as a model organism. And the premise of the study was to expose different groups of hamsters to different lighting schemes over the course of a month. So a control group just lived under a standard light-dark cycle with brightly illuminated days and dark nights, whereas the experimental groups were exposed to dim light every night, so about 5 lux of light, not a huge level, but with varying wavelengths. So 2 groups experienced white or blue light, which we know to be very potent cues for the circadian system. And then a 3rd group experienced red light, which we know is less potent. Okay, next slide. All right, so after about a month of living in these lighting conditions, we tested the hamsters in 2 different behavioral tasks that are meant to assess sort of depressive-like symptoms in rodents. So the first is a swim test that essentially quantifies escape behavior. So the idea is that the animals will initially attempt to find an escape from the pool, but over time they'll display helplessness and just kind of float until you take them out. And in pharmacological studies, mice that are given antidepressant drugs will try much harder to escape and not give up as easily. So that's kind of the basis for the test. In our hamsters, exposure to light at night increased the amount of floating, most prominently in the white and blue light exposure groups, suggesting that those animals had some altered behavior where they were giving up more easily in this task. The second test we did was a sucrose preference test, where the hamsters were offered regular drinking water or a sweet sugar water. And typically hamsters, like humans, will prefer to drink the sweet solution. So if they lack this preference, it's kind of interpreted as anhedonic-like response. Again, in our groups, we saw that exposure to the white or blue light at night decreased sucrose intake, although the overall amount of water drank across all the groups was the same. And so these results show that this very simple manipulation of exposure to light at night can actually alter behavior in a pretty strong way. We also asked whether light at night was changing the brain in any way. We know that neurons are incredibly plastic in response to stimuli, and we looked at the morphology of these cells to see if they were any different. Specifically, we counted the number of dendritic spines, which are sites of synapses on the cell. And they're known to change really rapidly in response to stimuli, and the changes in these have been associated with depression in people and things like that. So we saw some decreases in certain kinds of these dendritic spines in our hamsters that were exposed to white or blue light. But again, red light had a lesser effect. So we saw that light at night alters behavior, it alters neuronal morphology. What was actually happening, though, in the brain to elicit these changes kind of at a molecular level? So one factor, which Laura is really going to talk about in more detail momentarily, but which has been linked to depression in humans, is this neuroinflammatory environment within the brain. So patients with major depressive disorder have been shown to have increased expression of pro-inflammatory cytokines in the brain, which can alter neuronal circuits over the long term. Also, melatonin is known to have anti-inflammatory properties, and therefore its reduction with light at night exposure could potentially alter the neuroimmune environment. So for those reasons, we looked at the expression of pro-inflammatory cytokine genes in our hamsters after exposure to this dim light at night paradigm. And out of several candidates, we saw an increase in one of them, which is called TNF-alpha. And it's also been shown by many studies to be elevated in depression patients as well. Interestingly, the increase in TNF was rapidly reversed when we returned the hamsters back to just standard dark nights. And so we asked whether treating the hamsters with a TNF-alpha inhibitor could help prevent any of the behavioral changes that we observed. And so we did a study, and indeed we saw infusion of a TNF-alpha inhibitor into the brain during exposure to light at night actually prevented the hamsters from showing these sort of depressive-like responses in our swim test, providing a potential mechanism through which light at night could affect mood. So I'll let Laura go on now to tell you more about some of her latest research on circadian disruption and neuroinflammation and mood.
Okay, thank you, Tracy, for all of that wonderful information about the circadian system and introduction to neuroinflammation. So here I'll go ahead and transition the talk to looking more at disruption of the circadian clock and how it can alter neuroinflammation and lead to these changes in mood-related behaviors. So when most people think about affective disorders, changes in mood, the place that they immediately jump to is the brain, because we know the brain regulates our behavior. And the main cell that people think of in the brain are neurons, which makes a lot of sense because these are the main effector cells that drive our behavior. However, we know that there's also a lot of supporting cells in the central nervous system. So cells like astrocytes and oligodendrocytes are important support cells in the brain that can influence the way that neurons function. And my lab in particular is interested in how inflammatory cells in the brain, in particular the main immune cell microglia, and how microglia can mediate changes in behavior. And a common example of how microglia can regulate behavior occurs during sickness. So that's probably the most basic example of how our immune cells regulate behavior. So for example, when you encounter a virus like COVID-19, immune cells in your body get mobilized. But that signal doesn't just stay in the body. So immune cells get mobilized, they start fighting that infection, but they also send signals to the central nervous system to say there's an inflammatory response going on. So there's vagal afferents as well as active transport of inflammatory molecules called cytokines across the blood-brain barrier. Once your brain detects that immune signal, microglia, which are that primary immune cell in the brain, become activated, and they begin releasing inflammatory molecules into the brain. So cytokine synthesis is highlighted on this slide. And ultimately, what that results in is neural changes that are going to affect behavior. So if you think about last time you were sick, there's kind of stereotypical changes in behavior that occur. So the brain mediates things like fever when you're sick, but it also causes behaviors like social withdrawal, anhedonia, cognitive changes, lethargy, heightened pain sensitivity, etc. And these behavioral changes are actually very adaptive during the sickness response. So they help do things like conserve energy to recover from infection. They also help stop the spread of pathogen, for example, in the case of social withdrawal. However, these neuroinflammatory pathways that can also get disturbed in other situations outside of sickness, like, for example, with chronic stress or with aging, and result in long-term expression of these behaviors that are mediated by immune molecules in the brain. So basically, these pathways just aren't very specific, and they can get hijacked by any kind of immune stimulation, which may become maladaptive. So there's this literature that's demonstrated that in cases like depression, a subset of patients actually show increases in cytokines in the central nervous system, which may underlie some of the behaviors that we see develop. So linking this back to the circadian system, how does this relate to the circadian clock? Well, we know that immune cells have their own endogenous clocks that modulate the way that they function throughout the day. And importantly, microglia, that main immune cell in our central nervous system, have circadian rhythms in their inflammatory potential. So we showed a number of years ago now that microglia have much greater pro-inflammatory responses during the light phase in rodents, so their rest phase, versus the dark phase. So one of our questions, knowing that the circadian system regulates the way that these cells function, is, does circadian disruption induce changes in mood? Can those occur to dysregulation of the neuroimmune environment? And to test this question, we work with a RevHerb alpha knockout mouse. So Tracy already did a nice job introducing the circadian clock machinery. One of the secondary loops of the circadian clock involves this clock gene called reverb alpha. So it's involved in regulating BMAL1, one of those primary clock genes. And when you disrupt reverb alpha function, you actually delete the ability for that clock to have its 24-hour rhythms. So we hypothesized that circadian disruption induced changes in mood are due to changes in their neuroimmune environment. And we tested this question in these reverb alpha knockout mice. So first, we wanted to look at what happens to the neuroimmune environment in these reverb alpha knockout mice. And we specifically focused on the hippocampus because the hippocampus is a brain region implicated in mood-related behaviors, as well as it has a high density of microglia. And in our reverb alpha knockout mice, we actually see that there's increases in hippocampal microglia. Along with those increases in hippocampal microglia, we also see that there are changes in the inflammatory products that are produced in the brain. So in male and female reverb alpha knockout mice, we see increases in pro-inflammatory cytokine expression like interleukin-1 beta. And just to point out for the rest of the talk here, that little dots on the graphs represent individual animals, and females are always represented in purple and males are represented in green on these graphs. So we see increases in inflammatory cytokines with this reverb alpha knockout mice to a much greater degree in females as compared to males. So we see that reverb alpha knockout is inducing this change in the neuroimmune environment, but is it also inducing changes in behavior? Well, prior work from Colleen McClung's lab had highlighted that these reverb alpha knockout mice develop what's described as a mania-like phenotype. So they show changes in depressive-like responses, like Tracy showed earlier when she was going through data associated with light at night, so a more environmental circadian disruption. But it's also been shown that these reverb alpha knockout mice have changes in depressive-like behavior, as well as changes in risk-taking behavior, hyperlocomotor activity. And indeed, we replicated those findings in this study. So here I'll be showing you data from an elevated plus maze, which is the apparatus shown at the top left of the screen. So for this test, a mouse is placed in the center of this maze, and they can explore these closed arms versus the open arms. And in mice, because they're a prey species, they typically don't spend a ton of time investigating these open arms where they'd be more vulnerable to predation. However, they are exploratory in nature, so they are interested in engaging in some exploration of both these open and closed arms. And in general, in this assay, animals that spend almost no time in those open arms are considered to have an anxiety-like phenotype, and animals that spend a lot of time in the open arms may be thought to have be engaging in more risky or risk-taking behavior. So in our wild type control mice, so mice that don't have reverb alpha deleted, we see that they spend almost all of their time in the closed arm of that maze. So this is just a heat map from automated scoring software that looks at where mice spend their time in this maze. And you can see they're spending almost all their time in the closed versus the open arms. However, with our reverb alpha knockout mice, we see that they spend significantly more time exploring those open arms. So when we look at their preference index here, we see that they have almost no preference really for the open versus the closed arms. So we're engaging in this more risky, risk-taking behavior. And then they also show this hyperlocomotor phenotype. So when we just look at total activity in the elevated plus maze, we see that those reverb alpha knockout mice are spending a lot more time moving in this maze. And this is in contrast to their home cage environment. They don't, in general, have more locomotor activity when they're in a non-stressful home cage type environment. So this is really stimulated by placement in this novel apparatus. So from these data, what we can conclude is that these reverb alpha knockouts have changes in mood-related behaviors. And we did look at more than the elevated plus maze. This is just for time. I'm highlighting one assay here. And they also show this increase in neuroinflammation. So our next hypothesis was, are these immune cells in the brain really causal in inducing this change in behavior? So we decided to deplete microglia from the brain and look at how that affects the neuroinflammatory environment and these behavioral changes that are induced in that reverb alpha knockout mice. So to test this question, we work with a CSF1 receptor inhibitor that rapidly depletes most microglia throughout the central nervous system. And the CSF1 receptor inhibitor can be consumed in diet. So we place mice on a diet with the CSF1 receptor inhibitor. And after 2 weeks, we see that there's massive depletion of microglia from the hippocampus. So you can see microglia are labeled here in red and green, and you can see there are many more cells in animals that are on this chow diet versus on the CSF1 receptor inhibitor diet. So microglia are depleted about 90% overall. So what does this do to the inflammatory environment in the brain? Removing the main immune cell, we would hypothesize that this should completely, for the most part, eliminate those pro-inflammatory signaling molecules like interleukin-1 beta from the brain. And indeed, that is what we see in our male mice. So when we deplete microglia, you can see that there is almost no IL-1 beta expression in the hippocampus of male mice. Somewhat perplexingly, however, and this is why it's very important in basic research, people were a little late to the game studying females, but when we look at our female mice, we still see that our reverb alpha female mice have significant expression of that pro-inflammatory cytokine. So it is reduced overall compared to that control condition, but they still have this increase in cytokines, even though they had similar levels of microglia depletion. So we're not quite sure where that extra inflammatory signaling is coming from. It could be from astrocytes or another immune cell entering the brain. Like females tend to have higher levels of T cell dysregulation, so it could be another immune molecule. But our next question was, well, if we see changes in this neuroinflammatory environment, we've eliminated microglia, we've reduced cytokines completely in males and to some degree in females. So do we rescue some of these behavioral changes that we see in our reverb alpha knockout mice? And just to show with just microglia depletion alone, we don't affect the hyperlocomotor activity in our wild type mice. And in our reverb alpha knockout mice, we do see somewhat of a rescue. So they have a reduction from those animals on the control diet in that locomotor activity. And when we look at that more risk-taking behavior, so you'll remember in the elevated plus maze, we see that the reverb alpha knockouts don't show much of a preference for the open versus the closed arms. And in our male microglia depleted reverb alpha mice, we get a partial rescue of this behavior. So they look more like their wild type controls. However, our female mice that did not show that significant or did not show as great a reduction in inflammatory cytokines still, even with microglia depletion, do not have a reversal of that behavior. So overall, what we showed here is that depleting microglia partially rescues changes in mood induced by disruption of the circadian clock gene reverb alpha. And it did so to a much greater degree in males compared to female reverb alpha knockout mice. So this study really showed that microglia are necessary for some of these behavioral changes induced by circadian disruption. And in the final study that I'm going to talk about here, we kind of asked the reverse question, which is circadian disruption in microglia alone sufficient to induce changes in mood? So our next hypothesis was, does disrupting the microglia clock, so only the circadian clock in microglia, alter risk-taking behaviors in mice? So to accomplish this, we worked with microglia-specific circadian knockout mouse. So we work with a microglia-specific promoter here, TMEM119, and we deleted the clock gene BMAL, which is that counterpart to reverb. So they're involved in that same feedback loop. And we did this in adult mice. So it was an inducible knockout. And then we looked at changes in behavior after disrupting the clock, specifically in microglia. So first, when we look at that locomotor phenotype in the elevated plasmase, we see overall that disrupting BMAL in microglia induces that hyperlocomotion that we also saw in that global reverb alpha knockout mouse. And when we look at that change in preference for the open arm, so that risk-taking behavior, we also see that BMAL-specific microglia knockout mice show an increase in preference for the open arms. And that's just highlighted in these heat maps here. So taken together, what we can conclude from this study is that disrupting the circadian clock in microglia alone can lead to these changes in mood-related behaviors. So overall, changes in the neuroimmune system that are induced by circadian disruption may underlie some of the behavioral changes that we see that develop with disruption of the circadian system. So now, to transition back to thinking about how this affects humans and what are some of the strategies that we can have for rescuing circadian disruption. So as Tracy so elegantly introduced, we know that circadian disruption is really widespread in modern society with people that engage in things like shift work or transmeridian travel. But circadian disruption really affects almost all of us. So, 99% of the population goes home and after it's dark, turns on lights in their house, uses computer screens, etc. So I just have one more slide here to talk about some of the strategies for mitigating the disruptive effects of light at the wrong time of day. When Dr. Bedrosian and I started working on light at night in the Nelson lab back in 2009, there really wasn't much awareness surrounding light pollution. But since our initial studies were published, there's been a big increased awareness in the public about the effects of nighttime light exposure on things like sleep, mood, and more. So many of you are already probably aware of some of these ways that you can protect against light at night. So in recent years, smartphones come with an automatic app that kicks in at night and shifts the screen color from blue light. So most white LEDs are enriched in blue light, which, as Tracy demonstrated, those blue wavelengths are more disruptive for the circadian system. So a lot of phones now come preloaded with the ability to shift towards like a night screen at night, which is more red light enriched so that you don't get the same disruptive effects on the circadian system. People have also started using smart lights in their homes. So this is something that I use in my own home, and with my kids, we have hue color light systems, which you can control on your phone. So during the day, we'll keep our lights set to bright white or blue light. And then as the evening transitions, we'll shift those lights to a more orange spectrum of lighting. Shifting all the way to red can look a little bit scary, but as you get more towards that orange spectrum as well, you're filtering out a lot of those blue wavelengths of light. And then there's also things that you can purchase, like glasses that specifically filter out blue light. And there's been there was actually a JAMA article that came out in the last couple of days looking at melatonin supplements in the US and elsewhere. Melatonin is strongly suppressed by using light at night, and a lot of people have started to try to compensate for that by pharmacologically taking melatonin, supplementing with melatonin. Although, as the JAMA article that came out in the last couple of days shows, it's important to be aware of what you're taking, as some of these supplements aren't as exact as they should be. So with that, I'll just say thank you all for your attention. And I just want to highlight a few people that contributed to the work that I discussed today, including the lead authors on the studies that I talked about, Dr. Rachel Chen, she just finished her dissertation, and Dr. Louise Ince, and then and funding agencies as well. And then I'll pass it back to Tracy to do her thank you.
Yeah, thank you all for your attention. I'll just highlight most of the research that I talked about was performed in the Nelson Lab at Ohio State during my graduate studies. So I'd like to thank all of the collaborators on that work, as well as members of my current lab. We're happy to take any questions.
I have a question. Oh, Dr. Mason, you go first.
Yeah, I was just giving thumbs up. There's one question in the chat, and then a few other people just raised their hand. So I'll go ahead and share the one in the chat, and then we'll jump in for everyone else who's here. The one question was, would night lights be disruptive, or is the lux level low enough to not disrupt?
Yeah, that's going to probably depend on the individual nightlight. But, in the studies that we did with those hamsters, it was very, very dim level of light, kind of comparable to at least what I think of as a typical nightlight level. And we know that, in people, that melatonin gets suppressed so easily with, just a few lux of light. And so, I think there have been studies that kind of advise against sleeping with night lights in general for various reasons, including sleep disruption and other reasons. So I'll let Laura add anything if she would like.
They do make night lights now, though, as well, that are that longer wavelength of light. So you can get night lights that are more that deep orange to red that should be less disruptive. So it's kind of a complex question to think about, because in humans, unlike rodents, are nocturnal, so their eyes are open when they're exposed to light at night. When we stay up late watching TV, obviously our eyes are open, we're getting exposed to light at night. But when we sleep, we have the added that our eyes are closed. Some light still gets in through the eyelids, so people have shown that does occur. But and you have waking during your sleep cycles where you might open your eyes. But it's kind of a complicated question to think about.
We just put in this new nightlight thing in our bathroom, and it does have this orange red tone to it. And yeah, actually, when I get up in the middle of the night, I think it's kind of less disruptive to me personally.
I know there's quite a few folks in the room who had some: Carmen, Cation, Melissa, and then Laura. So whoever wants to jump in, go for it.
I have a question that has perplexed me for a long time. It's in the beginning of your presentation, you reminded us about the nightmare of being in the hospital for a few days. And it's amazing to me that we would think that the medical profession and, treatment in hospitals would be very aware of the connection between circadian rhythms and interrupting meaningful rest for the people who are sick. Yet, at least the people I know, the thing that is the worst is that you just get interrupted all the time. You don't get it's not just about the light, it's about noise and humans coming in and all of that. So I don't understand it. It's as if they either don't care or their priorities are different than trying to consider that extra variable of health and helping their patients get better. I just don't get it. Is it because it's so hard to run a hospital? Maybe.
It's actually, that's a really great point. And that is kind of what led the Nelson lab when we were in grad school and getting into this research question. Is one of the lab members had been had a staph infection and was in the ICU, and they complained that the lights were on all the time and like that couldn't be good for health. So that's why we started working on this. And at the time, there was almost no work except in shift workers, like nurses, on what light at night was doing to people. The only two groups I can think of are the shift workers themselves, and then there were a few studies looking at preterm infants, but almost nothing else out there on lighting in the hospital settings on patients. But since we started working in this area, it's really been exciting to see that there is a lot more awareness now. So when Tracy and I were leaving Ohio State, they were building a new heart hospital, and they made it like a circadian smart hospital where there was different light spectra at night. And I can say anecdotally, I've been in the children's hospital, actually, that Tracy works at Nationwide Children's, with my daughter who had surgery. And in the children's hospital now, they also have like more smart lighting and things to try to make improvements in this area. I don't know if Tracy has anything to add. She's the one in a hospital, so.
No, I mean, I think it's always a trade-off with, the safety concerns and allowing the staff to do their jobs and do what they need to do, getting in there at night. But, I agree, there's more of a shift towards, thinking about the strategies we can use to do the job that needs to be done, but also, help protect the sleep and circadian cycle of the patients.
Thank you.
So I have some comments and questions. I have some comments on just what you're talking about in the hospitals, because for one thing, it's quite interesting, because another technological area is the whole idea of how cost has been reduced and quality and kind has been increased in terms of cameras. So there's a lot more cameras that are able to monitor in not light, that could effectively and be financially probably feasible for installing in hospitals, so that they could monitor clients without monitor patients without light, because it probably was cost prohibitive before to have such kind of photographic monitoring. But it might be interesting to see how things go if these studies, if people start adopting the information you're finding from the studies that, about the disruption to sleep and how it can be, avoided. But the interesting counterpoint to that whole thing is hospice. when hospice comes to a house where someone who is, you know, kind of at the end of life, if they're waiting for family members, they want to give family members the most time with that person. Hospice personnel will leave the lights on all night, as bright as they can, to try and keep that client stimulated from passing on right away. So they're obviously aware, you know, that, and using that in a way to be advantageous to again the family members and so forth, having as much time as possible with the person before the total end of life. So that's another aspect to it. But my question is, you know, from a psychological mood standpoint, and I'm going to make a big leap here, but what my personal evidence is that and experience is that persons who have certain disorders, such as schizophrenia, for example, bipolar disorder, the things along that line will tend to automatically reverse day to night. They'll be up all night and they'll sleep all day. So I'm wondering how that plays into what you're telling us about this circadian rhythm. And, you know, is it a situation where their body is, because their body and brains are working differently, they really do need to operate the way they are? Or if we, you know, with the evidence you're showing, would it help them in some form to force the issue of reversing them back to sleeping at night and being awake during the day? So I have a question, you know, I'd like to know what your guys' thought is on that situation.
Yeah, I mean, the disruption, the circadian rhythm disruption is very well characterized in all of these, you know, kind of psychiatric disorders. It's like a prominent feature in all of them. It's kind of like, yeah, you're asking kind of chicken or egg question, I guess, in a way. Like, I guess it's, I don't know that we know the answer, really.
Okay.
You know, I mean, we know it's some, you know, like I mentioned, seasonal affective disorder, it's not really the same thing, but you know, in that case, the bright light therapy to sort of reset the rhythm and re-entrain it can be helpful for the symptoms. I don't know if we know that for other disorders like schizophrenia or manic depressive disorder. Do you know anything about that literature, Laura?
Well, bright light therapy is also effective in non-seasonal depression, but yeah, it's like Tracy said, it really is kind of a chicken or egg question, because circadian disruption is a key feature of many neuropsychiatric disorders. So for example, in major depression, you also get changes in like lengthening almost of the circadian clock, where people tend to stay up later at night, sleep later in the day. They also show suppressed cortisol rhythms. So typically we have this really robust rhythm in cortisol expression. And with major depression, it's not even always that cortisol is higher, it's more that the rhythm is completely flattened. So they're not getting the same signal. But it's hard to determine how that change in the circadian system is contributing. I know in my own research now, we study aging and Alzheimer's like pathology. And something that's interesting is in that case, often circadian disruption precedes other aspects of the disease. So there's this idea that it may be kind of like an early signal, but and then how does it lead to that disease pathology? Maybe it's through disrupting things like the way immune cells are functioning or other components.
Yeah, and you were talking about so it's the interesting is the cortisol part there, and the idea of the melatonin, and I was wondering on that also. So there's a thing, HTP 5-HTP, I think is what it is. It's the hydroxytryptophan, which I thought was a precursor to using in your body for to something along the lines of melatonin, but everybody's focused on more of the melatonin. So I didn't know, have you heard of this other kind of 5-HTP versus melatonin in terms of trying to do that regulation of circadian rhythms?
Well, serotonin is a precursor for melatonin on the synthesis pathway. So that they are related, and serotonin also is strongly regulated by the circadian clock. So when you have serotonin release or higher levels of serotonin from the dorsal raphe nucleus in your brain, it's very strongly controlled by the circadian system too.
Yeah, great.
I know we're at the top of the hour, so I don't know, Dr. Bedrosian and Fonken, I just wanted to thank you. If you all have to run away and do wonderful things in your research labs, thank you once again. If you want to stick around, we welcome you, but I didn't want to put that pressure on you.
Okay, well, I guess I'll jump off then. Thank you so much for inviting us, and it was great to talk with you all.
Yeah, thank you so much for having us today.